Virus-bacterial interactions can be important for successful viral infection and stimulation of the host immune response. But currently, highly concentrated and purified stock of human norovirus cannot be produced in the lab. This technique can be used with any virus that binds to bacteria for which either virus-like particles or live virus are available. It enables us to quantify interactions between these viruses and bacteria.
Inoculate 5 milliliters of liquid medium with a single isolated colony of Enterobacter cloacae from an agar plate directly from frozen glycerol stock. Grow the bacteria overnight. The following day, transfer 1.3 milliliter aliquots of the culture into 2 separate 1.5 milliliter centrifuge tubes. Centrifuge the tubes at 10,000 times G for 5 minutes. Remove the supernatants. Then, wash the samples twice, using 1 milliliter of sterile 1x PBS for each wash. Centrifuge the samples again at 10,000 times G for 5 minutes. Remove the supernatant and resuspend the pellets in 1.3 milliliters of sterile 1x PBS. Beginning with 0.5 milliliters of washed culture, serially dilute the bacteria in 1x PBS from 10 to the minus 1 to 10 to the minus 4. Using a spectrophotometer, measure the optical density of the washed undiluted culture and each of the 4 dilutions at 600 nanometers. Perform 10-fold serial dilutions in 1x PBS for each of the previous dilutions. Spread 100 microliters of the last 3 dilutions for each series onto